So the first of the two scientific programming interns is Ritvik. And what is something great about Ritvik is that from the minute I saw his resume, I had no doubt that he had all the skills to succeed in this project. He is even, I didn't even know, did you know that there are even programming competitions? And Ritvik is doing and winning programming competition. And the best thing that I experienced with Ritvik during this internship is to see how truly happy and excited he was to finally apply his programming skills, not just to competition, but to a real scientific problem, and to be able to help with these skills to study social memory. So something so far away from what you usually think about programming, but he really did great. So thank you so much for joining the internship and helping us.
Hello, my name is Ritvik Teegavapu. I'm a rising senior from American Heritage Boca, Delray, and my project at the USUDA Lab was to utilize computer learning for separation of fluorophores based on their spectral profiles. The overarching goal of the USUDA Lab was to link specific neuron activity that we were observing in vivo or live with social behavior that we were observing externally. On the left, you can see images from here from an actual in vivo mice, and right now it's in black and white just because of the imaging of the software. You can see here, this is the duplicated image from Amy's presentation, but it relies on the same fact that we're using a GRIN lens for deep brain imaging inside the brain. Now on the left here, we have the prelimbic region of the prefrontal cortex. The reason we chose this region in specific was because there's less neuron clusterings or densities here for us to easier sort the neurons. On the right-hand side, we have different fluorophores or fluorescent genetically encoded proteins. Now these are unique because we can excite them with specific light and they'll emit a different wavelength of light. You can see here that we've colored our specific neurons for a reason because we want to observe which functions they perform in the brain. As an example here, we have imaging neural activity. We have the GRIN lens placed in a deep brain imaging and with calcium labeled cells. Calcium is an important ion for neuron activation because it's prevalent when activating or sending signals. But you can see here that we're not able to tell the difference between any of these specific neurons. With the addition of fluorophores now, we have each individual neuron labeled with a specific color, still the same exact setup, but in this case we can pinpoint which neurons are correlated to which behaviors. Now, plotting all these fluorophores on an emission spectrum, we see this huge, wide range of fluorophores, specifically 11 that we're going to be experimenting this time. As you can see here, we have from blue, we're ranging all the way to red. Now, our experiment is going to theoretically hold like a bucket, and then each of these little wavelength markers, we're going to hold that bucket and see how much of each light is being put and placed in this bucket. But the problem here is that we have overlapping spectrums. We can see here, for example, sapphire and EGFP are essentially overlapping spectrums. This is problematic because we don't know how to differentiate between this light with our eyes, let alone, so now we've tasked the computer to do this. So previously, imaging has been done with co-registration, which is green and red. You can see here that the green and red spectrums are very far apart, and they're very easily discernible. We are now trying to do it with 11 fluorophores, which is problematic. But previous imaging that we've been doing has been firing all lasers at the same time, which in order to observe the fluorophores. We've now started to roll out on singular laser excitation. So you can see here that we're firing a laser of 405 nanometers. This is what the spike correlates to, and it only leads to a certain distribution of fluorophores remaining. You see here that tag BFP2 and turquoise are both here prevalent, and so is sapphire, but the remaining papaya, orange, they're all disappeared. So we only get a certain select subset of fluorophores to observe. Similarly here, we excite at a wavelength of 488 in this case. We see a lot of sapphire in the EGFP, and we also get a lot of the red, but all of the blue that we were previously observing has now disappeared. So we can continue on looking at individual lasers, 6 in total, and you can see that we're getting various subsets of fluorophores. Finally, one thing to notice here is that after we get to laser 6, which is a wavelength 639 nanometers, we're only left with one fluorophore, which is Neptune 2.5, which is interesting. Now, where does the computer science part come into this? We now rely on something called clustering algorithms, which is a different type of machine learning. But we should understand what machine learning is before getting into this. Machine learning is the ability for a computer to extrapolate based on previous data and determine patterns. Clustering algorithms in specific are trying to correlate patterns between different sets of data. For example, if I gave you 3 white balls and 3 green balls, you would immediately know how to cluster them based on their color. However, the computer can't do that. We haven't given it labels. We haven't given it any way or metric to sort them. So we need to be able to train this data in order to cluster it specifically for this. So the first part of the computational process is to reduce the noise by downsampling. Each of these pixels now contains some value, but however, there is some background associated with each of these. This is problematic because this is only a 4x4 pixel, but our images are almost 1,000 by 1,000, which is a lot of pixels to look at. So what we do here is we average the pixel noises and reducing it for the overall system. You can notice that we haven't changed the size of the pixel, we've only reduced the noise per pixel. The next step was removing the brightness feature. So in previous imaging, we were seeing here that in a regular raw image, there were super bright neurons firing, as well as the background, which was completely dark. However, looking at the cluster result, you can see here that it picked up on the pretty bright one, but it didn't really do anything with the background or contrasted it with a separate color. This is problematic because we don't want brightness to be a part of the thing. We only care about the specific colors, and we can't tell the difference between these neurons. We just know that they're super bright. So how did we get around this? We chose to normalize the data. So you can imagine here that this lower spectrum that I've drawn tracing out is of a darker pixel, and this lighter pixel belongs to this higher spectrum. We've essentially dragged up the lower pixel to the higher distribution, so everything remains at the same brightness. This therefore removes the brightness as a previous feature of this, and the computer can no longer use it to classify or distinguish between pixels. Finally, we reduced to the Frankenpixel, for lack of a cuter name. We now had to create a map of data with known pieces of 4-force. Why this is important? Well, machine learning heavily relies on training data. If we don't have a subset of data we know for sure was going to cluster where, it becomes impossible for us to train the computer in order to deal with testing images. So what we've done here is we've taken all these different 404 data that I've labeled here, and we created the quote-unquote Frankenpixel, which is essentially a stack of images that we know are going to predict how to cluster. So we know the red is going to cluster as the first thing, yellow is the second, etc. This also reduces the amount of size that we're having to deal and put in the computer. Why this is important? Because we were previously imaging 1,024 by 1,024 by etc. A huge amount of dimensions. Reducing this becomes a lot easier, since you can imagine that it's a lot easier to visualize something in 2D space as it is in 3D space. So by creating this one array, we still maintain the original image, and we can successfully move on to the clustering. Successfully might be a wrong word given the table, but we can see here that it pretty much picked up on all the Neptune clusters or pixels, all the Scarlet pixels, and all the Venus pixels pretty well. Then you get to the first 3, and it becomes an abysmal accuracy rate. The problem here is that we know that the clustering algorithm didn't work efficiently. That's true. What we do know is we have a theoretical reason as to why it didn't work. Previously, when we were normalizing data, we were removing pixels that were background or noise. The problem is that when you're clustering, it doesn't know what to do with that missing data, or it doesn't know how to make up for that. So in some cases, it's missing data that could potentially belong to one of these clusters, and the accuracy doesn't know, or the accuracy rate goes down as a result. So as a summary of the work, we wanted to take our raw image, downsample the noise per pixel so we wouldn't have to deal with it when fine-tuning the machine algorithm later, normalize our brightness so the computer could no longer classify it as a feature and remove the effect of the colors, create the quote-unquote Frankenpixel to first have a known set of data so we could train our machine algorithm on, and finally cluster, albeit unsuccessfully, our colors. Now the next step is that we have multiple clustering algorithms to employ here. The current one that we were using was the k-means, which is the most standard, and the one we'd used in previous research, as so we wanted to continue with that. But there are various clustering algorithms that we could use, and with that we can then determine which efficiency rate is the best and use it to definitively prove that we can co-register and express 11 colors at the same time. Thank you to my mentors, Dr. Mary Phillips and Dr. Nikolai Urban, for helping with my project, Dr. Larry Drago for creating this opportunity for all of us, the UCIDA Lab for being so accommodating and a nice environment overall, MP5 supporters for assisting with internship logistics, and my science teacher, Dr. Iris Thompson, for helping find my passion for research. I will now take any questions.
Thank you, Ritvik. Any questions? Don't you want to know more about Franken? I say Frankenpretzel, sorry.
The Frankenpixel, Is it physically stacked as you showed it?
No, that was just for visualization purposes, but as you can, oh wait, it's not there, but it's more stacked along, so each 404 is stacked in a row, and then we've labeled it down. So we've said the first 100 pixels of a given 404 go down the row, so there's 10 404s. So it's 1,000 rows for each 100 pixels, and then each of the 404s has a set amount of data along the column. So it's not visually stacked, as I said in the image, but for visualization purposes, it shows that we're stacking the data in a more compressed way than before.
Thank you.
So is the Frankenpixel like a euphemism for flattening the array?
Correct. And instead of having a three-dimensional array with our lasers all along the third dimension, we've now essentially flattened the array to serve as our training data for our algorithm, while also reducing the size of our array. So yes, flattening the array is a more technical term.
Hey, Ritvik, throwing you a curveball here. You said that you're using your algorithms to take the colors that look very similar, and you're going to use different excitation lasers to tell them apart. So after hearing Alana's talk, where she said she has different dyes that are hard to tell apart, and she could possibly tell them apart with different excitation lasers, do you think it would be possible to take some pictures with her samples and stick them to your algorithms and to try to tell the dyes apart?
Yeah, so the beautiful thing about machine learning is that algorithms are very adjustable. So there's a lot of things that we can back-end and develop to account for certain possibilities. So with overlapping dyes, we could create a theoretical probability distribution and say how much of one color is present in a pixel, and we could determine which of the colors is more prevalent based on it.
Great talk.